Well, thank you very much for the introduction. I do come from a cerebellar background from my postdoc. Unfortunately, I'm not going to talk at all about the cerebellum today, but it's a set of questions that I started thinking about when analyzing cerebellar data. So in my mind, there is a connection. So I'd like to start by first acknowledging the people who worked with me on this project. So both of the two projects I'll discuss were projects from Arthur Pellegrino, who is a PhD student in Edinburgh with Angus Chadwick, who's a collaborator of mine. So the first project, Arthur and Heike Stein, who's a postdoc in my lab, were collaborating, and for the second project, it was with Arthur and Angus. And Angus is looking for a postdoc, especially for the second part of the project that I will present. So if you're interested in that, please send an email to me or to Angus. And I'd like to start from the point of view that now we can record from very large populations, tens of thousands of neurons over very long periods. And the very common way of analyzing this high dimensional data is now to look at use methods like PCA or other dimensionality reduction methods to identify low dimensional latent variables hidden within the data. So this is an example of neural activity space. R1, R2, R3 are the activities or the firing rates of the three different neurons in this example. And methods like PCA look for low dimensional subspace here spanned by the two latent variables S1 and S2, in which the high dimensional activity is embedded. And the reason that we do this is not only because we love dimensionality reduction, but also because these latent variables, their geometry often has some information about the particular task that's being performed by that neural circuit. And so it seems important to develop and use these methods so that we have more and more clues as to the function of these different regions. And the question that I became very interested in while analyzing cerebellar data, because it's an area that's very important for motor learning and motor adaptation, is how these latent representations evolve over the course of learning. And while I was thinking about this, I started to realize that there are a lot of dimensionality reduction methods. There are a lot of methods specifically for neural data too, but there aren't as many methods that are specifically for uncovering latent representations in neural data over the course of learning. And so this started to pique my interest. And I will talk about two projects as much as I can in the short time frame. So the first one, they're both, the first one's on bioRxiv and the second on arXiv. So the first one really starts from going back to the idea of methods like PCA, which is a matrix factorization methods. What that means is that you take the data matrix, which here is neurons by time and has all the neural activity in it. You rewrite that or approximate it as the sum of R components of rank 1. So each of these components or latent variables is a rank 1 matrix. That means that it can be expressed as the outer product of one vector per component that corresponds to the neural encoding weights, and another vector that corresponds to the scores, or how that pattern, that population mode changes over the course of time. So this underlies PCA, but also NMF. A lot of different dimensionality reduction methods are based on these matrix factorization methods. And then when we want to look at learning, one thing we could do is really just make a very long matrix and look, use PCA on a matrix where we're just concatenating all of the different trials together. But really, in order to make some kind of separation between the time within a trial and the time across trials, what we can do is to stack those data matrices together into a tensor. So this is now a 3 tensor, neurons by time by trials. So just to get this out of the way, of course, you may have to do some kind of warping of your data, time warping, if it's not already, if the trials aren't all the same length. So we're gonna, for the rest of the talk, we're just going to assume that you've already warped your data, or it just fits in a tensor. Okay, so we've got here a method called tensor component analysis. This is a paper from Alex Williams, where you now have essentially an extra vector corresponding to each of these different components. And that vector corresponds to the weights over trials. Here you can see that in this example that I give, the weights are increasing slowly over trials. And so we could say that this is a component that corresponds to some latent variable that's being learned from trial to trial. So it's related, and looking at the shape of this third leg where we say that this is a component that's related to learning. So the limitation of TCA is that it makes an assumption, which is a strong assumption, that the latent dynamics are only changing in amplitude over trials. So you only really can have a change of this latent variable according to this gain, the amplitude of this particular loading vector here in trials. But you wouldn't be able to have a change of the temporal profile from one trial to another, for example. And it turns out that not all neural variability can really be described by this type of view where you have low dimensional latent representations of the neural activity in some fixed subspace. So one example is if your neural encoding vector changes, so maybe it slowly drifts over the course of days. I'm giving the example of representational drift, but it could be slow changes as a result of learning, for example, or it could be condition-specific changes of the encoding vector. And another example, which is much more classic, is just neural sequences, which are not described by a single, zero-lagged population mode that increases and decreases in strength over time. And to try to capture these types of variability, we need to start looking at a different way to think about the data tensor. So there's a paper from 2016 that already noted this fact. And what they saw is that if you take the data tensor and you unfold it into a matrix, you can unfold it in different ways. So you can concatenate trials, or you can concatenate neurons in order to turn this three-dimensional tensor into a two-dimensional matrix. And different regions are actually better described by different unfoldings of the tensor and then applying PCA on them. And so in order to capture all of this type of variability, we need to go back to the definition of these types of methods. So we start with a rank 1 tensor, which is how we define TCA. But it turns out that you can define different types of ranks for tensors. And one that's more recently been discovered by Terry Tao is the slice rank. So here, instead of each tensor, so on the left, a rank 1 tensor is the outer product of 3 vectors, but in a slice rank, the slice rank 1 tensor is the outer product of a vector and a matrix now. And so the difference is that, for the mathematicians out there, the difference is that this matrix is no longer constrained to be rank 1, as it is on the left. Okay, and then of course, depending on which of the 3 dimensions of the tensor is used for the loading vector, there are 3 different types of slice rank 1 tensor that you can imagine in your decomposition. And what we've tried to show is that each of these different slice rank 1 tensors are actually capturing different types of covariability. So the first one, which we call the covariability across neurons, is capturing some kind of characteristic neural weights with temporal dynamics that can change in an arbitrary way from trial to trial. So this is the typical type of component that you get when you apply PCA in the normal way that we usually do. However, if you now look at the component type where the loading vector corresponds to trials, this is now picking up characteristic trial weights, but the temporal dynamics can be heterogeneous from neuron to neuron. So now you can capture things like sequences, neural sequences, or like a temporal basis function in this particular example. And then the final class captures shared variability across time. In other words, you have a loading vector that corresponds to characteristic temporal dynamics, but the neural encoding weights can change from trial to trial, and we think that it could potentially pick up changes like representational drift that could happen on a slow time scale. And so what we've are proposing is to essentially fit the data tensor into a low slice rank approximation. So now you have three different types of components that correspond to these three different classes. And so we call this sliceTCA because it's similar to TCA, but using the slice rank of the tensor. And just to make this very explicit about the relationship between sliceTCA, TCA, and PCA. So if you just took one of these components and you unfold this matrix into a very, very long tensor, if you only fit one type of component, you're just doing a matrix factorization if you ignore the other types of component. But the strength of sliceTCA is in disentangling all of these different classes at the same time. So if you consider these different classes of covariability in this little Venn diagram, so doing PCA on each of the three unfoldings would correspond to shared variability across neurons, time, or trials, each of these three different circles. Here I have a picture here for two examples. sliceTCA is capturing the union of all three of those different types of covariability classes, which could be mixed in the same data sets. And TCA is actually capturing the intersection of these classes. Because if you think about a tensor rank 1 component, that can be expressed as any of these three different slice rank 1 tensors. So TCA is really in the intersection of these three different covariability classes. Okay, so in the interest of time, I'm going to show two different applications to data. So the first is to a motor cortical data set, which is from Mark Churchin's lab. This is in the neural latence benchmarking website. So essentially, it's recordings from PMD and from M1 of a monkey that is performing a reach task, but it's a little more complicated. So there's barriers, and the monkey has to make a curved reach around the barriers. So you have the maze condition in which it makes these curved reaches, and then there's a control with a no maze condition where it makes a straight trajectory to the same goal location. Okay, so here's what the hand positions look like on the straight condition and the maze condition. And if you try to decode these hand positions, so actually we're decoding the velocity and then we're converting it to the hand position. If you do the decoding based on the raw activity, but after trial averaging for each condition, so each of these curves is the average decoded trajectory for that particular condition, then you see that it matches very well to the actual hand position. But that's only if you do trial averaging. If you take the raw data and you try to do the same decoding analysis, you can see it's much, much noisier. So the next thing you might think is maybe that's because there's all this variability that we can discard that doesn't contribute actually to the behavioral dynamics. And one way that we can do that is doing something like matrix factorization, like PCA. Since the data is non-negative, I'm actually going to do NMF instead of PCA, but similar idea that I'm only taking here 12 components from NMF, redoing the decoding based on these 12 components, and you can see that actually the decoding is much worse. If you instead use TCA, you do a lot better already. But you can do something that's actually even more simple, which is to do NMF, but on the other one of the other unfoldings of the data tensor. So now this is the trial slicing NMF, and you can see that you can actually decode much, much better than any of these other cases. And here there's no sliceTCA, there's no fancy new method. This is just doing NMF on a slightly different matrix that comes from the same tensor. Okay, so the reviewers also asked us to compare against LFADs, and it turns out that you actually do about as well with sliceTCA as with LFADs. In the case of the maze condition, you actually do a little bit better. Here, this is the R square, so you do a little better with sliceTCA than with LFADs. So you don't necessarily need to go to a very complicated method in order to tease apart this information. So then the next question that we ask is, what do we get by adding an extra slice type? Because in what I showed you before, that was just looking at NMF on a different matrix. So here we have the decomposition that I showed in the previous slides. What we're going to do is add a single component of a different slice type. And specifically, I'm going to add the time slicing. And the reason is because these are recordings from M1 and from PMD. And we expect that premotor cortex has preparatory information, which is known to have information about the upcoming movement, and which we expect to have some kind of characteristic activation immediately before the onset of movement. So we expect that because there should be some characteristic time course, we'll look for it with the time slicing component. So these are what you get from the trial slicing component. So here I'm just showing the trial factors. You can see that they're very tuned to specific reach directions here in the no maze condition. They all look like this, so, have with tuning either in the no maze or maze condition, and they all have these sequential dynamics. We did cross-validate the sequences to show that so this part at the beginning is not real. That's just an artifact of sorting. But right around here, this is real in the sense that if you cross-validate, you get the same sequence corresponding to the same component. But if you look at a different component, you see that you don't have this same structure anymore. So the trial slicing components are picking out condition-specific sequences that seem to be associated with the movement dynamics, because they're happening right around the period of movement here. And then we look at the time slicing component, and here's the loading vector, and you see that there's large activation immediately before the time of movement, which is at 0, and then it shoots down. And if you look at this slice, which is trials by neurons, and you analyze it, so you can actually decode the end position from this slice in line with what you expect from preparatory movement that should have information about the upcoming goal points. And what we did is look at the representational similarity of these encoding vectors. So now we're looking at trials by trials, and here they're colored by the different reach trajectories. And if this is in premotor cortex, and you can see a lot of structure here because the encoding vectors for similar reach directions are similar in PMD. However, if you look at M1, you see that correlation structure disappears. And we think that's because premotor cortex has this extra preparatory information, which is signaling the dynamics of the upcoming movement. And then we also show that the fraction of variance explained is a bit higher in the PMD subset of the data than in M1. So together, this we believe shows that the time slicing component is finding some kind of information about motor preparation, whereas the trial slicing component is showing information really about condition-specific sequences that are related to the movement dynamics themselves. In the interest of time, I'm not going to go over these practical issues, but I just want to allay your fears that we did think about the practical issues involved in fitting this. So one of them is, how do you choose the number of components? So we have a very rigorous cross-validation procedure that we implement and that we describe in the paper. Another issue is how to identify a unique solution. So it's known that in matrix factorizations that there is an invariance to rotations. So in the context of sliceTCA, that would look like, say you have this tensor that you can write as the sum of two neuron slicing components and a time slicing component. The problem is we can now, if we stack together these two components together, right, and we're going to unravel this slice into a long vector. So now we can write it in the same way that we would do for PCA, the weights times the scores for this particular slice type. The problem is that we can insert an invertible matrix here, and then we can rotate the weights and rotate the scores. And so now you no longer have a unique solution to this decomposition. So this is an invariant transformation that is inherited from from matrix factorizations, and we can use matrix factorization methods to take care of this. And then the second invariant transformation is that it turns out you can actually transfer a rank 1 tensor between the slice types. So I'll go through this quickly. You can read more about it in the preprint. But if you have two components of different slice types, what I can do is add and subtract a tensor rank 1 tensor that has the same loading vectors of these two different components, and then it has a third loading vector that is arbitrary. And then what I do is I pop one into the first slicing component and one into the other one, and then I can regroup by these shared loading vectors. And so now I have two different ways of rewriting the same, exactly the same tensor with different slices. And so this is an invariant transformation that's specific to sliceTCA. And I won't go through it, but we did think about it very hard, and we came up with a way to find basically a multi-stage optimization procedure that comes up with a unique solution. So we first, after we find the decomposition, we essentially place a regularizer on the subtensors of each slicing, which takes care of that second invariant transformation, and then we use matrix factorizations to take care of that first invariant transformation. But it's all described in the preprint if you're interested, and I'm happy to take questions on this too. So we have this this pipeline for the analysis that we thought very hard about. Okay, so going back to the idea that different regions might actually prefer different slices. We tried to tackle this question in a data set from the International Brain Laboratory. So these are neuropixel recordings from many different regions during a perceptual decision-making task. And I'll show you all of the components that we find with sliceTCA. So we get two trial slicing components that we find really seem to capture behavioral variables. So here we see the first component is capturing correct in orange versus error trials in black. And if you look at the activations of these different regions, so thalamus, APN, and MRN, you see there's a strong activation at the time of reward. And it seems to pick out the correct trials. So we were worried that this could be related to some artifacts somehow of how we were warping the data. So we actually looked into the original PSTHs to see if we could find any examples. And we do see some. Here's an example from APN. There are some PSTHs of single neurons that seem to have a very sharp activation at the time of reward only for correct trials, but not error. So that made us feel a little more confident that it's not some kind of spurious signal. The second component has a non-monotonic shape, which correlates statistically significantly with the reaction time. So it has a correlation of about 0.3, I think, and is statistically significant. So these two trial slicing components seem to capture features of the task. When we look at the neuron slicing component, so here we found three different components. So the first, and they clustered hippocampus, dentate gyrus, and visual cortex. So in this first component, we see that it's mostly active in the hippocampus. And then we actually found that if we looked at the slice, we found that there was an increase in activation with contrast, which we didn't necessarily expect from the hippocampus, but we did see a few examples of that in the in some hippocampal neurons that you see here in the PSTHs. We also, saw the fourth component seems to pick out the dentate gyrus with a suppression of activity following reward. And the fifth component seems to pick up the visual cortex. And then if we look at the time slicing components, they seem to pick up specific time periods of the task. So component 6 is picking up the period around reward. Component 7 is picking up the time around the cue, and component 8 is picking up very late times in the task post reward. And this is just to show that we thought that maybe these were somehow like summing up to become a flat baseline that was being fit for the different neurons, but it actually turns out that at least for about half of the neurons, it's statistically significantly different than just being a flat baseline. Okay, and then the last thing I wanted to cover was about sliceTCA was what these actually mean, because we're used to thinking about low dimensional representations in the neural space. And because of the symmetry of the decomposition, so like the trial slicings would correspond to low dimensional representation, but in some vector space where each vector, each axis corresponds to a different trial. And then even more insane, the time slicings would be like a low dimensional representation where in a space where every axis corresponds to a different time point. So we wanted to translate everything back to the neural space, which is how we interpret neural activity for purposes of representations and decoding. And so I have a few schematics to show you how to think about these different types of covariability. So first, the neuron slicing is the easiest because it's very closely related to how we think about usual PCA applied on neural data. So we have some neural activity that's constrained to a low dimensional subspace, but within that subspace, it can have any particular temporal profile from trial to trial. The time slicing component, because you have characteristic temporal profiles, it has the same temporal shape, but that shape is embedded in a subspace that can now change from trial to trial. So here I'm showing a rotation of the low dimensional subspace, but the dynamics within the subspace has the same characteristic shape. So you see it starts to become less low dimensional because now the low dimensional subspace can change from trial to trial. And then the trial slicing components actually can be high dimensional because they can capture things like sequences. And so you have high dimensional dynamics that can not really be described as being embedded in a low dimensional subspace, but we can still capture them because they have some characteristic change from trial to trial. And then of course, all three of these are combined to make the entire reconstruction of the data, which can look more complicated than any of these three at the same time. And so we think this is exciting because sliceTCA is maybe a way to have a method that's quite mathematically interpretable, but edge it away from the low dimensional geometric view of neural latent representations. Okay, so to summarize this project, we show that sliceTCA is able to capture and demix these three different covariability classes, and that the behavioral information is distributed across different covariability classes, which is why it's important to think about them, and also that the different covariability classes are differentially expressed across areas, which is why we think it could also be important going forward with multi-region recordings. Okay, so in the last part of the talk, I will discuss a different project that kind of grew out of sliceTCA, where we wanted to more directly tackle the question of learning. So in sliceTCA or in TCA, these decompositions, we can say that it captures learning if the trial leg is increasing slowly over time. So I think I have that little here. Yeah, so here's just matrix factorizations again. So here's a low rank approximation. So this is TCA. And if we see that the weights are kind of slowly increasing over time, then maybe we'll say this is due to learning. But it's not really directly addressing the question of learning. And part of that, I think, is because we don't really have a generative model for the dynamics. What we can observe from neural activity that we record is the geometry of the representations, but the geometry of the representations are shaped by the dynamics. So in other words, they're shaped by the vector field of the neural population that is determining how those trajectories will look, right? So here's an example where we have some straight trajectories that are shaped by a particular vector field. Over the course of learning, those trajectories will change in geometry. So now they become curved, but that's because the vector field is changing in some way in order to shape the geometry of the trajectories. And so we became interested in trying to more directly tackle learning by asking, how can we infer how the vector field is shaped over the course of learning? And in the case of neural dynamics, we know that one of the main contributors to the vector field is the recurrent dynamics, of course. So here's an example of what the weight matrix might look like for a particular neural region, right? So we have the neuron by neuron weight matrix. This is on the first trial. And if we think about it in terms of learning, on each trial, we'll have a different weight matrix because it's slowly changing. But, of course, when you start working with tensors, then you see tensors everywhere. So what if instead of just having these side by side, we put it together into a tensor? And so now we have what we're going to call the weight tensor, which is neuron by trials. And we can analyze how that weight tensor changes. And so what we started thinking about is, instead of doing a low rank decomposition on the neural data tensor, what if we constrain the weight tensor to be low rank, similar to TCA? Of course, we still want to fit neural data, so we're still using the neural data tensor, but the constraint now is going to be on the weights and not on the activity. And then we can use RNNs as like some kind of generative model for how we get the neural activity. So this is the big picture. And then, yeah, so we look at again, we're looking at the changes in the trial factors. And if we see some slow increase, then that gives us an idea that it could be due to learning. And actually, we're going to specifically constrain these trial factors to be smooth by essentially making them correlated from trial to trial, similar to Gaussian process regression. And that's going to find trial factors that are smoothly changing from trial to trial, so that we really can, get to this question of how the vector field is changing over the course of learning. So RNNs, of course, are often used to be trained to perform tasks, but you can also use them to fit neural data. So you have some generic RNN that receives some input U of t on a particular condition that's fed in through some weight matrix B. And instead of having a linear readout that is then some variable that's performing a task, we have a readout that is approximating the neural data matrix, or in a couple of slides, it'll be a tensor. So this is not our idea. There are other papers that have taken this approach, but this is just to remind you that we can use RNNs to fit neural data as well as fitting performing tasks. Okay, so the RNN dynamics are shaped by the weight matrix. And it's known that there's a special class of weight matrices that are very convenient because they are tractable, and we can use them to understand neural computations. These are low-rank RNNs developed by Francesca Mastrogiuseppe and Sergeant Osteic. And the idea is that if you have a low-rank constraint on the weights, so I'm going to write that low-rank constraint in the same way here, just in different forms. It turns out that then what the low-dimensional subspace is, in which the dynamics that control the trajectories are embedded. So you can see that by taking a generic equation for an RNN, we just pop in this low-rank weight form here into the weights, and then what we can see is we group together the B transpose and then the phis, the activations. Now you have a dot product here, which is just going to give you a scalar value that's multiplied by the column vectors in this weight matrix. So what that means is that to step out and take the big picture, is that with low-rank RNNs, the reason they're so useful is because we know a priori that the x's, which are the membrane potentials, are going to lie in a low-dimensional space that's spanned by the AIs and also the column vectors of the low-rank decomposition of the weights, but also the column vectors of the inputs. So this becomes very useful then, because we already know what the low-dimensional subspace is. We don't have to go looking for it. And so the way that we connect this to learning with is to make this constraint that the weight tensor is low tensor rank. So this is the full model. We have a low tensor parameterization of the weights. So we're forcing them to be low tensor rank. And again, we're constraining the trial factors to be evolving smoothly over trials, because we want it to capture the changes due to learning. So the variability as a result of learning, and not the variability as a result of the conditions. Instead, the conditions are coming in through the inputs that we're also fitting. So again, the learning related variability is all being funneled into the weight tensor, and the condition specific, the condition related variability is being funneled into the inputs. Okay, and then what's really useful about low tensor rank RNNs is that at every trial, it's a low rank RNN. So for every trial, you can write it as an outer product of these two vectors with some scaling factor. So that means that it inherits all of the nice things about the low rank framework from Francesca and Surgeon. But in addition, because the C's, these components are just being changed in amplitude from trial to trial, actually it's not just that at every trial it's low rank, it's that it's the same low rank structure over the entire experiment, which means that if we do the same mathematics, I'll go through it quickly because of time, but what that means essentially is that we know that it's the same low dimensional subspace over the entire course of the experiment. And we're identifying that by fitting it to neural data. So if you tried to fit this data by having a different low rank RNN on different trials, there's no guarantee that you could compare the results from one trial to another trial. So here, by making this constraint, we know that all of the dynamics and the neural representations are going to be constrained to the same low dimensional subspace from trial 1 to trial 100. So that's really what's useful about this. And then we can see how that affects the changes in the geometry of the representation. So I'll give you an example. This is, we just have some sinusoids, so just a toy model. We start with a ring of initial conditions, and then we feed it through the RNN that's constrained to be low tensor rank. This is the low dimensional space that we know a priori, and we know it's not going to change through the experiment because, again, it's of the low tensor rank constraint. And this is just showing you an example visualization of how we can visualize the changes in the geometry of the neural manifold across the entire experiment in the same latent subspace. So this is why we think this is very, very exciting, basically, to be able to show these changes in neural data. Okay, so let's see. I'm going to wrap up by showing a couple examples from the same motor learning paradigm. So this is data from Matt Parrish, where there is a monkey that is learning to move a joystick towards a position on the screen. He's controlling the cursor. And here's the baseline. So he's moves the cursor. In the perturbed trials, they add a force perturbation to the joystick, which causes the monkey to make these curved trajectories, which then straighten out over the course of adaptation. And then in the washout, they remove the force field, which causes it to first start to rotate in the other direction and then eventually straighten out. So the first thing that we did, so first, this is not data. This is, we first just trained an RNN of unconstrained rank, so just like a regular vanilla RNN with no constraints, to perform the same motor task. So here's the error in the perturbation, the washout, and here's how the RNN solves the task. Because we want to analyze the changes in the dynamics of the RNN, where we have the ground truth weights. So I'm not going to show it because of time, but we validate that the weight updates are low rank and low tensor rank. And then what we do, I think this is really cool. So if you look at the eigenvalues, so blue are showing the eigenvalues, and then as we go into the perturbation block of trials. Those are in pink. And then in blue, again, here, this is showing the washout. So you see all these random eigenvalues that aren't doing anything for the task because they don't change at all over trials. But there's only a few of them that actually do seem to be important. And what you see is that they start real, they become complex during the perturbation because the RNN has to make these rotational trajectories, right? And then during the washout, the rotation has to go away. So it goes back to the real line. So that gives us some intuition for how this task-trained RNN is actually performing that task by looking at the eigenvalues as they change over trials. What we did is to fit a low tensor rank RNN to the same data. And you can see that what it does is take all these random eigenvalues that are not corresponding to the task performance, and it shrinks it all to 0. And what's left is really just these, this conjugate pair of eigenvalues that seems to be responsible for that rotational change in the perturbation and the washout. So we thought this was really cool. Okay, so now we're going to apply it to data, and the data that I was describing, which is PMD and M1 recordings from Matt Parrish in these monkeys during this motor task. First, we just show, that we're able to capture a lot of the data with low tensor rank RNNs. So here, this is the cross-validated unexplained variance for LTR, our model with different numbers of hidden units. And you can see that it reaches this minimum value around rank 5. So it does seem to be very low rank. This gray value is what is the cross-validated unexplained variance that we get if we fit all of the entries to the tensor. So we can't really do better than that. And you can see that it reaches that performance in just a handful of components. So it appears to be low tensor rank. If you're wondering why we're not able to do better, keep in mind that we're not capturing, there's nothing in the model that captures trial to trial variability. It's just capturing variability due to learning and variability due to condition. If you're interested in the trial to trial variability, you should send me or Angus an email, because this is what we'd like to extend in the future. Okay, and so this is what you get if you just fit a low rank RNN that has no component that's changing over learning. So this 5% difference is attributable to changes that are due to learning. And so what we can do now is look at the trial factors and see that it captures transient and sustained effects of the adaptation. So you see this is in the perturbation block of trials. You can see that the geometry of the initial conditions seems to capture the target angle. This is something that you can do with PCA, of course, but it's not nearly as clean and doesn't show you this beautiful ring-like topology. And you can visualize the geometry in the latent subspace, which again, we know a priori, and it's the same for the baseline, the perturbed, and the washout because of the low tensor rank constraint, where you don't see so much from PCA. And you can also infer changes in the vector field and changes in the eigenvalues. Interestingly, we see different, it's not solving the task in the same way as the task-trained RNN. As you can see, the eigenvalues are different, and it looks like the changes are very small, which we'll just have a shout out to Barbara Fulner's paper. We think it might be, if you know this paper. But in the interest of time, I'm going to wrap up with this last slide, showing the same changes in the manifold structure that I showed you before for a toy model, now for this data. So here you can see that what happens on the perturbation block, there's going to be a rotation right now here of the entire manifold. You see it rotates. And then it's going to, at the end of the perturbation right now, here, it rotates back. So this is the change that we saw in this particular data set. I'm going to show it again. So it rotates right now. You can see it rotate there. And then at the end, you'll see it rotate again. Down here. Yeah. So this is the change in the manifold structure that we saw in this particular task, which I'll admit is not as cool looking as the sinusoids, but we're interested in continuing further and to analyze how the geometry and the dynamics change over learning. Okay, so to summarize, we have a model that parameterizes the RNN weights to be low tensor rank. And by making this constraint, we're able to capture changes in learning through the changes in the dynamics, which we can now infer, and we can also capture changes in the manifold geometry in this data set. If you're interested in, in these ideas, but not necessarily interested in the methods, there's a review that I wrote recently that you might be interested in. And I think that's about time. Thank you.